Hi, and welcome to RadOnc Talks, a lecture series designed for students and residents of radiation oncology. My name is Ria. I am currently a PGY 5 resident at the University of Pittsburgh, and today we will be covering another high-yield radiation biology topic by defining some very important terms, including OER, LET, and RBE. OER stands for oxygen enhancement ratio, and it basically describes how well does oxygen help a particular type of radiation work better. As we will find, some types of radiation work the same whether or not they have oxygen present, and some types of radiation work better under oxygenated conditions. So we'll kind of define what the oxygen enhancement ratio is and talk about various factors that influence OER, including different types of radiation, stage of the cell cycle, the oxygen concentration, and talk about different types of hypoxia, acute versus chronic, as well as markers of hypoxia that we can use to kind of measure how the oxygen enhancement ratio is taking effect. LET stands for linear energy transfer, and it's kind of exactly what the term sounds like, which is how much energy is transferred over a particular linear distance for different types of radiation. So again, we're going to talk about the definition, how it differs for different types of radiation, and connect it back to the OER and talk about the relationship between LET and OER for types of radiation. And then we'll end by discussing the RBE, which is the relative biological effectiveness. And again, we'll define what that is and how it differs based on various factors, including the type of tissue we're dealing with, the fraction size of radiation, and then tie it back together with the terms that we defined earlier, including LET and, you guessed it, OER. I think that OER, LET, and RBE are difficult concepts. They took me a very, very long time to understand, and it took me even longer to be able to apply these concepts to test questions. The best way, I think, to study radiation biology is really to do test questions, but my goal with this video, again, I'm not an expert in radiation biology, but my goal is to explain a very foundational understanding of these concepts so that when you go on to do test questions, hopefully things start to make sense. Before we jump into defining these terms, I do want to just review some basic concepts in radiation biology. First, we know that there are different types of ionizing radiation, right? There's indirectly ionizing versus directly ionizing radiation. Indirectly ionizing radiation are uncharged particles. So these can be your photons on the electromagnetic spectrum, such as X-rays and gamma rays. These are massless particles that result in the release of electrons when they interact with our atoms. And then there are neutrons, which have a huge mass, much, much heavier than electrons even. But neutrons are very heavy particles that can interact with the nuclei of our atoms and result in the release of protons and alpha particles. So indirectly ionizing radiation is anything that's uncharged that interacts with our atoms and causes the release of charged particles. That's where that indirectly ionizing term comes in. Directly ionizing, then, are any particles that are charged. So electrons, protons, alpha particles, carbon ions, they themselves have a charge, and so they're considered directly ionizing. When ionizing radiation interacts with our bodies, the way that it works to kill cells is by causing DNA damage. So the DNA damage can be caused directly by those ions, or it can be caused indirectly by the interaction of those ions with molecules such as water, resulting in the formation of free radicals. And then it's those free radicals that go on to indirectly damage the DNA. So there is direct versus indirect ionization, and then there is direct versus indirect DNA damage. Now, the next thing that's important to understand is how the radiation causes DNA damage. Radiation can cause all kinds of DNA damage. It can cause base damage, it can cause single-stranded DNA breaks, it can cause double-stranded DNA breaks. When we think about how DNA damage results in cell kill, it's really those double-stranded DNA breaks that ultimately lead to cell kill. The reason for this is that double-stranded DNA breaks are the hardest for our cells to repair accurately, because if we have single-stranded DNA break, remember we have DNA which is two complementary strands that are bound together. And so if we just have one side of that broken, then the complementary strand is a nice template to fix it. So single-stranded DNA repair and base damage, all of that stuff can be easily repaired, but double-stranded DNA breaks are the hardest to repair. When the DNA has replicated in preparation for cell division, so that happens at the end of S phase. S phase is our replication phase, and late S, G2, we actually have 2 replicated strands of DNA and all of the protein machinery that we need to fix damage. It turns out that that is when the cell is best at repairing double-stranded DNA breaks, because again, even if we have a double-stranded DNA break, now the entire chromosome has been replicated, so we have a second kind of chromosome to work off of, and then we can better repair those double-stranded DNA breaks. So this process is known as homologous recombination, and because homologous recombination is highly accurate and faithful, the cell turns out to be most radioresistant, so meaning it's hardest to kill cells when they are in the late SG2 phase, because now they have that extra power. They've replicated the DNA, and they can repair double-stranded DNA breaks most accurately. So these kind of fundamental concepts we have gone over in previous lectures, and if you need more details on that, I would recommend watching the additional videos in the radiation biology playlist that I've created in RadOnc Talks. But these concepts are very important to understand, fundamentals of radiation biology. And then everything that we talk about in radiation biology really builds on these basic concepts. So when we talk about OER, LET, RBE, we're basically just further understanding the different types of radiation, how they work in our body with or without oxygen, and then how we can manipulate various factors such as fractionation, dose per fraction, number of fractions to better kill our cancer cells with radiation. So with that fundamental background in mind, let's start talking about oxygen enhancement ratio. Earlier, I said that oxygen enhancement ratio is basically a measure of how well oxygen helps the radiation to work better. That is a very basic way of defining this term. The actual definition, the textbook definition of oxygen enhancement ratio, is the ratio of the dose that's administered in hypoxic conditions to the dose that's administered in aerated conditions to achieve the same biologic effect. So think about that. The denominator is the dose that we have to give under aerobic conditions. The numerator is the dose that we have to give under hypoxic conditions. So if oxygen is playing a huge role in helping the radiation to work better, then we're going to have to give less dose in aerobic condition, decreasing the denominator, and we're going to have to give more dose in hypoxic conditions to get that same effect, thus increasing the numerator. So therefore, the higher the oxygen the oxygen enhancement ratio, the more that oxygen helps with cell kill. So higher OER means that oxygen helps more with cell kill. Now, how does oxygen help with cell kill? What oxygen does is it actually fixes or it makes permanent the damage that is caused by free radicals. So free radicals damage DNA, and when oxygen is present, it makes that permanent. And remember, when we're giving our indirectly ionizing radiation, such as X-rays, gamma rays especially, about 2/3 of the damage that we cause is due to indirect damage caused by free radicals. So oxygen really helps to fix that into the DNA, and it helps make that damage permanent. So without oxygen, the indirect DNA damage that we cause, especially by X-rays and gamma rays, can really be repaired. Now, different types of radiation rely to a different extent on that indirect DNA damage, and therefore on the oxygen enhancement effect. For sparsely ionizing radiation like X-rays, gamma rays, and even protons, these are considered sparsely ionizing, and they do rely a lot on oxygen to fix that permanent damage caused by free radicals. So the OER for X-rays, gamma rays, and protons is somewhere around 2.5 to 3, which is pretty high. That tells you that under oxygenated conditions, the radiation damage by X-rays, gamma rays, and protons is around 2.5 to 3 times as high as it is under hypoxic conditions. The more highly ionizing the radiation gets, the less and less it relies on that indirect DNA damage, and therefore the less it relies on oxygen. So the oxygen enhancement ratio then decreases. So neutrons are considered intermediately ionizing, and I just think of them as being heavier, and so therefore they're more powerful, and they can just go and directly damage the DNA themselves. So the OER for neutrons is lower, it's about 1.6, so oxygen still helps a little bit. And then alpha particles are densely ionizing, and their oxygen enhancement ratio is actually 1. So think about what an OER of 1 means. It means that the dose administered in hypoxic conditions is the same as the dose administered in aerated conditions to get that same biologic effect. So that means that there's really no enhancement from oxygen. So 1 is the lowest possible OER, and OER actually cannot be less than 1. So the big takeaway here is that oxygen enhancement ratio tells us how well oxygen helps to make the radiation work better. For sparsely ionizing radiation, so X-rays, gamma rays, protons, these are the most common types of radiation that we use in clinic. Their OER is high at around 2.5 to 3. Neutrons are intermediately ionizing, so their OER is about half of that, 1.6. And then alpha particles are densely ionizing, and oxygen actually doesn't help them at all because they are so densely ionizing and they work so well on their own. So the OER for alpha particles is 1, meaning that there is actually no enhancement effect from the oxygen. The image shown in this slide here compares the survival curves for X-rays, neutrons, and alpha rays under hypoxic versus oxygenated conditions. And as you can see, with X-rays, the survival curve drastically changes with the addition of oxygen, so that is the blue curve. For neutrons, it's not as significant, and then for alpha rays, there's no change at all. So the OER for X-rays and gamma rays, and also protons, so these are our sparsely ionizing radiation, the OER is about 2.5 to 3. Neutrons, which are intermediately ionizing, the OER is 1.6. And then for alpha particles, which are densely ionizing, the OER is 1, meaning there's no enhancement effect from the addition of oxygen. Now, one important point is that in order for the oxygen enhancement effect to occur, oxygen has to be present either during or within microseconds of the radiation exposure. This is because whenever the free radical damage happens, the radical DNA can actually be restored to a sulfhydryl group. But if oxygen is present, then oxygen binds to the radical and quote unquote fixes it, meaning it makes it permanent. So the free radicals that damage DNA have a lifespan that's on the order of 10 to the negative 5th to 10 to the negative 9th seconds. And so therefore, if oxygen is going to help, it needs to be present either right when the radiation hits or within microseconds of the radiation exposure. Believe it or not, the OER actually differs in various phases of the cell cycle. Remember in our review portion, we mentioned that the cell is most radioresistant because during S phase, because that is when the DNA has been replicated. So it's really late S, early G2 that the cell is most radioresistant. Well, believe it or not, that radioresistant phase is when the OER is the highest. And then when cells are more radiosensitive, they tend to be most radiosensitive in the M phase and also during G1 phase. The OER actually is lowest during the G1 phase. And the way that I remember this is I think of it as the OER is high when oxygen can help the most. So if something is working well on its own, we already talked about our densely ionizing radiation, works really well on its own, so the OER is low. The radiosensitive part of the cell cycle, the radiation is already working really well, so oxygen has less to add. If something is working well on its own, then the OER is low. But if oxygen can help make things better, so if something is more radioresistant or more sparsely ionizing, that is when the OER stands to help the most. That's when oxygen can have the most enhancement. So the approximate OER for X-ray radiation during G1 is somewhere on the order of 2 to 2.5, whereas in S phase, it's around 3 to 3.5. So on average, the OER for X-rays is said to be around 2.5 to 3, but that really depends on which part of the cell cycle we're hitting it with. So if you have a hard time remembering this, just think about when things are working on their own already, then oxygen has less to add, so the OER is going to be lower. But if something is not working as well, so if we have sparsely ionizing radiation, or if we're dealing with the radioresistant portion of the cell cycle, then oxygen has more to add, and that enhancement effect, the enhancement ratio, the OER is going to be higher during those times. So hopefully now we understand what OER is and how oxygen actually helps certain types of radiation work better by fixing or making permanent that DNA damage. The next question is, what concentration of oxygen do we need to maximize our OER? It turns out that the relationship between the radiosensitivity and the oxygen concentration is actually logarithmic. So that means that initially we see huge gains as the oxygen concentration increases at low levels. We see huge increases in the relative radiosensitivity. And then after a certain point, those gains really decrease and we don't see much improvement as the oxygen tension goes up and up and up. So these are some important numbers to be aware of. The maximum OER that we see, so 100% oxygen effect happening, we see at about a 5% oxygen concentration, which is around 30 millimeters mercury. We see about 50% maximum OER concentration at 0.5% oxygen concentration, which is around 3 millimeters mercury. So you can already see that logarithmic relationship. So remember these numbers. 50% of the maximum OER is at 3 millimeters mercury, or about 0.5% oxygen. And 100% maximum OER is seen at 5% oxygen, which is 30 millimeters mercury, so 10 times that. And then above 5% oxygen or 30 millimeters mercury, we're already at 100% OER. So increasing the oxygen concentration above that does not further increase the OER. So biggest takeaway here is that there's a logarithmic relationship. We see the maximum OER at 5% oxygen, which is 30 millimeters mercury. And we see 50% effect at around 0.5% oxygen, which is 3 millimeters mercury. Now, we've talked a lot about what's happening at a really molecular level in terms of oxygen fixing the DNA damage caused by free radicals. But if we zoom out for a second, let's think about what is actually happening at a cellular level. So oxygen travels to cells via our bloodstream, right? And various blood vessels, I should say, kind of penetrate the tumor. And when we think about how oxygen diffuses, oxygen is carried through the blood vessel, and then the oxygen particles actually have to diffuse out of the blood vessel and reach our tumor cells. So when oxygen diffuses, it can really only travel to a maximum of about 200 micrometers from the bloodstream. The distance that oxygen diffuses is about 70 to 200 micrometers. So within that radius of the blood vessel is really what can be reached realistically by oxygen. So if we look at a tumor and kind of look at the relationship between the tumor cells and the distance from various blood vessels, we realize that there are certain areas that are acutely hypoxic, meaning there's a short-term hypoxia that is developed from a temporary closing or blockage of a blood vessel. And then additionally within the tumor, we also have areas of chronic hypoxia. So these are really, really far away from blood vessels, often due to permanent closing or blockage. Oftentimes, what just happens is that the tumor cells divide so rapidly that they rapidly increase their radius, so they increase their distance from that blood vessel, and there's not enough time for more blood vessels to grow kind of in the center of the tumor. And so we get these central areas of tumor that are often necrotic. They are areas of chronic hypoxia, and oxygen just can't reach there because the tumor has grown so big and we don't have a mechanism for blood vessels to kind of get in there centrally. So these areas of chronic hypoxia are just far away from the blood vessels in general, or they can be due to some type of permanent closing or blockage. So because there's no oxygen, these areas are often necrotic. They are often less sensitive to the radiation because they are hypoxic, right? And most of the time we're using sparsely ionizing radiation, such as either photons or protons. And so these areas end up being less sensitive to radiation, and that can limit the success of our radiation treatment. Now, it is possible for these cells to undergo reoxygenation, so areas of acute hypoxia can rapidly reoxygenate. Areas of chronic hypoxia are unlikely to reoxygenate, but sometimes they can slowly reoxygenate. And one of the reasons that fractionation of radiation works so well is that reoxygenation is one of the 4 R's of radiobiology, right? Each time you hit a cell with radiation, a lot of the aerated cells are going to die off and the hypoxic cells are going to be left alive. So some of those hypoxic cells might be chronically hypoxic, so they're difficult to reoxygenate. But the acutely hypoxic cells that are from that rapid growing of tumor can kind of then better access blood supply. And as the tumor shrinks with each dose, more and more acutely hypoxic cells are reoxygenated. And that allows for the oxygen effect to help those cells to die off from the radiation as well. So this is kind of important to understand, understanding that oxygen can only travel about 70 to 200 micrometers. And so the distance between the blood vessel and the tumor cells is really important to think about. And beyond that 200 micrometers is really where we're going to get these necrotic areas that develop from chronic hypoxia. And that's really the area that's toughest to reoxygenate. The acutely hypoxic areas can easily reoxygenate. In the Eric Hall textbook, they actually talk about markers of hypoxia and different hypoxic radiosensitizers. So there are certain markers that we can actually use to visualize hypoxic areas. So there are certain molecules called nitroimidazoles, such as pimonidazole, which is a hypoxic marker. And in the image here, the green areas are the hypoxic areas that are stained for pimonidazole. There's another transcription factor that's important to be aware of called hypoxia-inducible factor. This is basically a transcription factor that gets activated in hypoxic conditions, and it promotes transcription of genes that respond to hypoxia. So I think the mechanism for hypoxia-inducible factor is kind of cool because under normal conditions, when there is oxygen, it is hydroxylated. So it literally, when there's oxygen, think of oxygen binding to hypoxia-inducible factor. And that causes it to bind the VHL or von Hippel-Lindau component of a protein ligase called E3 ubiquitin protein ligase. So whenever ubiquitin is bound to anything in the cell, that kind of targets it for degradation via proteasomes. So under normal conditions, when there is oxygen present, the hypoxia-inducible factor is bound to oxygen, it's hydroxylated. That causes E3 ubiquitin protein ligase to bind and targets it for degradation. So when there's oxygen, oxygen binds the hypoxia-inducible factor and it gets degraded. But then when there's hypoxia, so no oxygen, that hypoxia-inducible factor is no longer hydroxylated, so then it's no longer degraded, and that allows it to go on and transcribe genes that respond to hypoxia. So there's something called HIF1 alpha, which is a particular hypoxia-inducible factor. And in this image, the red stain is showing kind of the gradient of oxygen. And you can see that as we approach those green areas of hypoxia, the red sort of increases. And that increases, more and more distant from those blue areas, which are the blood cells that are stained for CD31. So you can kind of use these markers, the nitroimidazoles and hypoxia-inducible factor, to visualize hypoxic areas of tumor. And then nitroimidazole is also important to realize because it's a hypoxic radiosensitizer. So these are just some markers to be aware of. Those were the most high yield factors I wanted to discuss regarding OER. Let's move on and talk about linear energy transfer, or LET, and then talk about how LET connects to OER. LET is the average energy transferred per unit length of a track, and it can be measured in kiloelectron volts per micrometer. So if we want to write out a formula for LET, it's literally the change in energy over the change in length. So how much energy is transferred per unit length of a track. And if we look at a very microscopic level for any given molecule, the energy per unit track length can kind of vary. And so there's actually two different ways of calculating the average. There's the track average and there's the energy average. So the track average basically takes set lengths along the track and counts how many energies are deposited in each segment. So let's say you're taking a distance of 2 micrometers, and every 2 micrometers you count how many energies have been deposited and you average that out. So you're kind of averaging out the numerator. That's the track average. The other way to do it is take the energy average, and you basically count every 4 energies and measure the track distance between every 4 energies. And as those track lengths change, you then average out the track lengths. So that's averaging out the denominator. So there's two different ways to calculate it, track average or energy average, and you're either averaging out the numerator or you're averaging out the denominator. But what is important to understand is LET is the change in energy over the change in length, and it's really the average energy transferred per unit length. So because it's an average, there's two different ways to calculate it. Track average, you average out the numerator. Energy average, you average out the denominator. Just as OER was different for different types of radiation, LET is also different for different types of radiation. And remember, LET is basically the change in energy over the change in track length. And as we move from sparsely ionizing to densely ionizing radiation, that LET increases, right? Because we get more energies deposited per unit track length as we get more and more densely ionizing radiation. So as a result, the LET is much higher for alpha particles than it is for protons, X-rays, and gamma rays. And it's also higher for neutrons than it is for protons, X-rays, and gamma rays. So this table just shows some example LETs, and let's start by looking at the different particles, right? So X-rays and gamma rays and protons, they're all on the order of something like 0.2 to 2 keV per micrometer, right? The 10 MeV protons are higher, they're 4.7 keV per micrometer, but we're talking like single digit LETs here, or even less than one LET. Alpha particles, on the other hand, the LET is 166 keV per micrometer. So the LET for alpha particles is much higher than it is for protons, X-rays, and gamma rays. That is a huge takeaway. For densely ionizing radiation, the LET is higher. Now, let's look at the energies. So if we look at cobalt gamma rays, the cobalt-60 gamma rays have an energy of about 1.1 MV. And then for 250 kV X-rays, the energy is 250 kV. So X-rays, this particular X-ray has a lower energy than the cobalt-60 gamma rays, which are 1.1 MV. So if we compare those photons, the 250 kV X-rays actually have a higher LET. It's 2 versus 0.2. If we look at protons, the 10 MeV protons actually have a higher LET than the 150 MeV protons. So the 10 MeV protons have an LET of 4.7, and the higher energy protons of 150 MeV have an LET of just 0.5. So what we're seeing here is that as we get more densely ionizing radiation, the LET increases, but within a given type of charged particle, the LET is actually higher at lower energies. So if you're looking at X-rays and gamma rays, the lower energy photons are going to have a higher LET. Same thing with protons. The lower energy protons are going to have a higher LET. I imagine this as kind of traveling through a field of tulips. So I think of it as higher energy particles kind of move faster through the tissue. And because they move faster, they're able to pick up less tulips. They can deposit less energy per unit track. The lower energy particles, on the other hand, move slower through the field of tulips, and so they have time to stop and smell the flowers and pick up more tulips. So the lower energy particles that are moving along in a more relaxed fashion have time to deposit more energy or pick up more tulips per track length. So that's how I remember. That's kind of the visual analogy I have for remembering that lower energy particles of a given type of particle have a higher LET. Not to be confused with the higher LET for densely ionizing radiation. So always, 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 alpha particles are going to have the higher LET compared to things like protons, X-rays, and gamma rays. But within a given energy proton or within a given energy photon, the lower energy particle is going to have a higher LET. And that is where that analogy of traveling through a field of tulips comes in. As you have higher energy within a given type of particle, it's going to move faster through the field of tulips and not have as much time to pick up tulips. But the lower energy particles have more time to stop and smell the flowers, so they pick up more tulips, or in other words, they deposit more energy per unit track length. If we look at the survival curve comparing X-rays, neutrons, and alpha rays, we can see that the LET increases as the shoulder of the curve decreases. So it always confused me, what is the shoulder of the curve? And I think it makes sense if you look at the X and Y axis. So in these survival curves, we're plotting the surviving fraction in the Y axis against the dose in the X axis. So with X-rays, the survival curve has a pretty large shoulder, meaning that initially in the curve, the slope is less than as we go further along the curve. Basically, that means that at 2 gray per fraction or 4 gray per fraction or 6 gray per fraction, we have less killing with each fraction compared to if you increase it to 6 to 8 to 10 gray per fraction. So as we increase the dose per fraction, the killing increases, and so the slope of the curve increases and fewer cells survive. So X-rays have a pretty large shoulder of the curve. But if we look at higher LET particles like neutrons or alpha rays, even at a low dose per fraction, the slope of cell kill is much higher. So for neutrons, it's higher than X-rays, and for alpha rays, it's even higher than it is for neutrons. So for these higher LET particles, even at a lower dose per fraction, the higher LET particles are causing more cell kill, even at a lower dose per fraction. And that's really what the shoulder of the curve is, right? The shoulder of the curve tells us how much the slope changes. So for these highest LET particles like alpha rays, there's really not a shoulder to the curve because the slope isn't changing a whole lot. With X-rays, however, which are lower LET, they are causing significantly more cell kill with greater and greater dose per fraction. So the slope is changing more, and therefore there's a higher shoulder of the curve. So that's just a visual depiction of this. For some people, I think this helps. For others, it doesn't. But the biggest thing to realize is that X-rays have the lowest LET, neutrons have a higher LET, and alpha rays have an even higher LET when we're comparing types of radiation. And because X-rays have a low LET, they have the biggest shoulder of the curve. Higher LET radiations don't have as much of a shoulder. Now, what happens to OER as LET increases? Well, let's think about it. We started by talking about sparsely versus intermediately versus densely ionizing radiations, right? X-rays, gamma rays, protons, these are sparsely ionizing. Neutrons are intermediately ionizing, and alpha particles are densely ionizing. We know that as we move to more densely ionizing radiations, the LET increases, right? Because we know that alpha particles have a higher LET than neutrons, and neutrons have a higher LET than X-rays and gamma rays. And then we also know that the OER was highest for our sparsely ionizing radiation because oxygen has the most help to give for the type of radiation that's probably the least effective. So oxygen effect ratio, or oxygen enhancement ratio rather, was highest for X-rays and protons at around 2.5. So the big takeaway is that as LET increases, as we get to more densely ionizing radiation, the OER actually decreases. So just to remember, the approximate enhancement ratios for X-rays and protons, OER was about 2.5. For neutrons, OER was about 1.6. And for alpha particles, it was basically no OER, which is an OER of 1. That's the lowest possible, right? So OER for alpha particles is 1, OER for neutrons is 1.6, OER for X-rays and protons is 2.5. And these are inversely related. So as the LET increases, the OER decreases. That brings us to our very last term that we'd like to define, which is the RBE, or the relative biological effectiveness. To me, the intuitive definition of relative biological effectiveness is basically just how potent is the radiation. What is the damage rating of the radiation? How biologically damaging or how biologically effectiveness is the radiation? That is kind of like the intuitive definition that you would think about if you just hear relative biological effectiveness, right? How well does the radiation work? Now, it's all relative, and the textbook definition, the consensus that has been decided for what is that measure against which to rate the effectiveness of any other type of radiation, is the dose of 250 kV X-rays. So that is just a consensus that somebody came up with, and it's what we've gone with, and that is how we define the relative biological effectiveness. So basically, the textbook definition of RBE is the dose of 250 kV X-rays to the dose of any other type of radiation that you need to cause the same level of damage. So RBE really is what we intuitively think about, which is how potent is the radiation? How damaging is the radiation? And then the way that we define it in a universal way is by saying, well, how much dose of 250 kV X-rays would we need to cause the same amount of damage as caused by any other type of radiation at any other dose? So when you think about RBE, think about what you're treating, what type of radiation you're using, and what type of dose you're using, and ask yourself, what is the dose of 250 kV X-rays that I would need to cause the same level of damage? And if that number is high, if you think you would need a higher number of 250 kV X-rays to cause the same level of damage, then your RBE is going to be higher as well. This, I think, is the hardest concept to understand. I definitely think it's the hardest, it's the toughest concept to intuitively understand. So we'll go through each of the different factors that affects RBE and try and explain how those factors work. But we need to realize that RBE can change with the quality of the radiation, or the LET, the dose of radiation, the number of fractions we're using, the dose rate, and also the biological system or endpoint, so what tissue we're treating. So I think the easiest way to kind of wrap our heads around this is to one by one go through each of these factors and explain how RBE changes and why. Let's start with RBE and the type of tissue that we're treating, only because this was one of the hardest ones for me to understand. So as we said, RBE is kind of a biological endpoint, right? It's how much damage are we causing to the cell. And think about how much DNA damage are we causing with our radiation. Remember, DNA damage varies from sublethal damage to lethal damage, right? And the lethal damage that we think about is double-stranded DNA breaks, but there's also a lot of sublethal damage that ideally can be repaired. When we accumulate more sublethal damage, so when we are worse at repairing the sublethal damage, the RBE is higher. Which tissues are going to be the worst at recovering from sublethal damage, do you think? Do you think it's the tissues that are used to proliferating all the time and constantly used to replicating their DNA? Or do you think it's the cells in our body that don't replicate a whole lot and the cells that are kind of matured and not really actively replicating? Which cells are more likely to accumulate sublethal damage? Or in other words, which cells are less likely to be able to have a capacity to repair sublethal damage? I think this is a hard question to answer, but the bottom line is that the cells in our body that are not used to replicating, so think of our late responding tissues like spinal cord, central nervous system, kidney, lungs, and so on. The cells that are considered late responding with a lower alpha beta ratio, these are the cells that are not as good at repairing sublethal damage. So they tend to accumulate sublethal damage more, and as a result, the RBE is higher in those cells. Early responding tissues like our tumor cells, skin cells, intestinal cells, these cells have a greater capacity to repair sublethal damage. And I think of it as they're simply replicating their DNA all the time. So they're constantly used to kind of fixing DNA errors. And so they have a lower RBE because they tend to accumulate less sublethal damage. If you have a hard time making sense of this, go back to the trick that I mentioned earlier. Remember, RBE is basically the ratio of the dose of 250 kV x-rays that we need to whatever other type of radiation that we need. So when we're changing the tissue type, ask yourself, what is the dose of 250 kV x-rays that we would need to cause the same level of damage in a given tissue type? Would we need a higher dose of 250 kV x-rays to damage our high alpha beta ratio rapidly proliferating cells? Or would we need a higher dose of 250 kV x-rays to damage our late responding tissues like spinal cord, kidneys, and lungs? And if you understand how late responding tissues work, you know that more x-ray dose is needed in a single dose fraction to cause greater damage to our late responding tissues. And therefore, the RBE is higher for late responding tissues. And if you want to understand why, it's really because those late responding tissues have less of a capacity to repair sublethal damage. If you understand the relationship between RBE and the tissue type, hopefully the relationship between RBE and fraction size will also make sense. So as we learned when we talked about RBE and different types of tissues, RBE is higher as we have greater accumulation of sublethal damage. As a result, RBE is actually going to get smaller as our fraction size increases. So RBE decreases with increasing fraction size. And why is that? With a smaller dose per fraction, we allow for more sublethal damage. And therefore, at smaller dose per fraction, the RBE is actually higher. And as we get to higher and higher dose per fraction, the RBE actually decreases. So this can be very complicated to understand if you overthink it. Just remember, RBE is higher with greater amounts of sublethal damage. And when do we get more sublethal damage? Number one, if we're treating our late responding tissues, so think low alpha beta ratio. And number two, if we're treating with smaller fraction sizes. So both late responding tissues and smaller fraction sizes are going to have a higher RBE. The most important relationship to understand, the most commonly tested thing, is the relationship between RBE and LET. And if you're going to memorize one thing from this entire talk, memorize this fact. RBE increases with increasing LET up to an LET of 100 keV per micrometer. Beyond 100 keV per micrometer, as LET continues to increase, the RBE decreases. So RBE increases with LET up until 100 keV per micrometer, and then as LET continues to increase, RBE decreases. Now, why is that? That's the million-dollar question, right? I had memorized this fact for a very long time, for probably 2 years of residency, and I don't think I understood why until less than a year ago. So it basically has to do with the LET of 100 keV per micrometer and the DNA diameter and their relationship. So the DNA diameter is about 2 micrometers, and at 100 keV per micrometer, the distance between ionizations is kind of optimized to cause a double-stranded break with a single track. Beyond the LET of 100 keV per micrometer, we actually get too many extra ionizations, and those ionizations get wasted. So we end up causing too many ionizations, more than what is needed to cause a double-stranded DNA break, and those ionizations get wasted, therefore lowering the RBE. So that is why we get increasing RBE with increasing LET, but only up until 100 keV per micrometer. And then after an LET of 100 keV per micrometer, you really have diminishing returns. So when the LET goes up beyond that, the RBE decreases, and it all has to do with the diameter of DNA. So when I understood that, it kind of blew my mind. But definitely memorize that RBE increases with increasing LET up until an LET of 100 keV per micrometer. And then after that, RBE decreases even as LET continues to increase. Now, the RBE of X-rays is less than that of neutrons, which is less than that of alpha particles of lower energy, as long as the alpha particles have an LET less than 100 keV per micrometer. And that makes sense because, remember, RBE increases with increasing LET up until 100 keV per micrometer. Another important fact to memorize is that the RBE of protons is about 1.1. So protons have a very similar RBE to photons, but their RBE is about 1.1. So whenever we're dosing with protons, we have to keep that in mind, that their RBE is 1.1. The exact clinical importance of that is probably a little bit beyond the scope of this lecture. Biggest takeaway here, RBE increases with increasing LET up to an LET of 100 keV per micrometer. And so that is why we see increasing RBE for alpha particles more so than neutrons, more so than X-rays, because as long as the LET is less than 100 keV per micrometer, the RBE is going to increase with increasing LET. But then after we exceed that 100 keV per micrometer, we're kind of in that area of diminishing returns where we're causing more ionizations than we need. So more ionizations than the diameter of the DNA. And so we're kind of wasting ionizations, thus leading to that drop in the RBE. Just in case I haven't said this enough times, remember, RBE increases with LET up to an LET of 100 keV per micrometer. And then beyond that, RBE decreases with increasing LET. And it all has to do with the number of ionizations relative to the diameter of DNA. When LET is greater than 100 keV per micrometer, we're depositing more ionizations than are necessary to cause double-stranded DNA breaks. And so we're kind of wasting energy, which results in decreasing RBE above a 100 keV per micrometer LET. So we've connected RBE to LET. Let's briefly talk about the relationship between RBE and OER, or really the relationship between RBE and oxygen. For high LET radiation, RBE can change with the tissue oxygenation. The reason for this is, remember the reference dose that we're using for RBE, right? When we define RBE, the reference dose in the numerator is 250 keV X-rays, which are low LET radiation, because X-rays are sparsely ionizing, so they are low LET radiation. And remember that low LET is subject to the OER, whereas high LET is not. And so for high LET radiation, that denominator is always going to be constant, but the numerator will change based on the presence of oxygen. And so if we're dealing with hypoxic cells where there's no oxygen, the RBE is going to be higher, right? Because it's high LET radiation, so that denominator of our RBE equation is constant, but we need more 250 keV X-rays to cause the same effect when we're dealing with hypoxic cells. So for high LET radiation under hypoxic conditions, the RBE is going to be higher. For aerated cells, however, the RBE will be lower because again, the denominator is constant for high LET radiation, but then the numerator is 250 kV X-rays. And when we have some oxygen around to help, in aerated cells, the 250 kV X-rays that we need are going to be fewer. We're going to need less dose of 250 kV X-rays. So that is why for high LET radiation, the RBE is really subject to tissue oxygenation. And it's because we have that dichotomy where the high LET radiation doesn't depend on oxygen, but the reference dose that we're using to define RBE, which is those 250 kV X-rays, that highly depends on the oxygen. So here you really have to think about the numerator and that we need more 250 kV X-rays in hypoxic cells. Therefore, RBE is higher when we have hypoxic cells. So this slide, I think, has the biggest takeaways that you need to remember from this talk, which is the connection between LET, OER, and RBE. So as LET increases, OER decreases, and we already talked about why. Approximate OERs for X-rays and protons are about 2.5, also for gamma rays around 2.5. For neutrons, it's around 1.6, and then for alpha particles, it's 1, which means there's no OER, there's no enhancement from oxygen. That's the lowest possible OER that you can get. When there's no enhancement from oxygen, the OER is 1, and that's what we see for alpha particles. And then RBE increases with LET until 100 keV per micrometer, and then it decreases as LET continues to increase above that. And that's because we're getting too many ionizations to cause double-stranded DNA breaks, more than what is needed, so we're essentially wasting energy. So LET increases as OER decreases, 2.5 for X-rays and protons, 1.6 for neutrons, 1 for alpha particles. These are approximate OERs to memorize. And then RBE increases with LET until 100 keV per micrometer, and beyond that, it decreases. And just to reiterate, I think it makes the most sense to think about OER, LET, RBE in very layman's terms and understand what they mean. But it's also really important to memorize the textbook definitions because that's what comes up on tests. And sometimes putting it in the context of textbook definitions really helps when you're answering multiple choice questions. So OER is the dose that you need in hypoxic conditions to have a given effect over the dose that you need in aerobic conditions to have the same effect. And if you need more dose under hypoxic conditions, then that means that the OER is high. So that's what we see for things like X-rays, gamma rays, protons, sparsely ionizing conditions. LET is the change in energy over the change in track length, and it's really an average change in energy over the change in track length. And LET also increases as we get to more and more densely ionizing radiation. And then finally, RBE is the dose of 250 kV X-rays to achieve any given effect related to the dose of any other type of radiation to achieve the same effect. So RBE is really a measure of how well does the radiation work, and the standard reference dose that we have decided is 250 kV X-rays. So if you can memorize these three definitions and understand the relationship between LET, RBE, and OER, I think that is a very strong foundation to start answering test questions correctly related to these important concepts. Again, I can never reiterate enough how much of a non-expert I am in this topic. So I really am boiling down these very complicated concepts to super fundamental foundations. And I understand that a professional radiobiologist may be able to explain these a little bit better and maybe with more detail and accuracy. My hope here is really to just define these terms in a way that makes sense and build a foundation that helps to start taking practice questions and answering those correctly. Again, if you're studying for the radiobiology exam, I do think the best way to prepare for this is really to do practice questions. There's great questions available online through the Astro study guides, also through Radon questions. So doing more and more questions on these topics really helps to reinforce the concepts and understand the different ways that they can be asked about. Hopefully in this talk, I've been able to kind of hone in on the textbook definitions of these terms, highlight some of the important relationships that are important to realize and how those come up. And I hope that that helps as you move on in studying and start to answer some practice questions. So good luck with studying. I hope this was helpful, and thank you so much for your time and attention.